So in the last segment, we finished up talking about codons and their implications in protein synthesis. All right. This illustration sort of depicts the value of codons. If we look, we see that here's your template, or here's your double-stranded DNA. This particular strand is now going to serve as a template. So these strands are separated. Then the information in this series of bases is converted to messenger RNA. So A pairs with U, C with G, and so on and so forth. So here is the RNA that is made. Now this is unedited RNA, so we just take what comes directly from the DNA here. Now what we see is that these bases are read, again, in triplets, or 3 bases at a time, in codons, or as codons. And again, each codon specifies a particular amino acid, whether it's phenylalanine, glycine, serine, whatever the case may be. Now, of the 64 possible triplets, most of them code for amino acids, but there are 3 that serve as stop codons. So there are particular codons that when they are read on the ribosome will not add an amino acid to the chain. They will signal to stop the production of the polypeptide. Let's see. Another important idea here is the concept of a reading frame. There are particular groupings of these bases for correct ordering of the amino acids. So the particular reading frame dictates the correct way to read this messenger RNA sequence to produce the polypeptide. Now, if you insert a base, or delete a base, let's imagine we delete this G, then that would make the codon, the first codon, UG and then U. Well, UGU could produce a different amino acid than UGG. In the same way, if you add a base, like if we added an A here, that would shift the reading frame over. So changing the reading frame almost always leads to deleterious or harmful effects. Last concept, there is redundancy but not ambiguity in the code. Now, here's what that means. If we look at this code chart, this is the kind of chart you'll need to read on the test. First off, let's look at how to read this. Let's say we have a sequence or a codon AUG. Okay, oh boy, oh, he's acting up again. Okay, so let's say we have the sequence AUG. If we look, the first base is read along this side, so there's the A, so we know the amino acid is going to be in this row. The second base is U, so we go down and go to that column, so now we have this, now we know the information is in this area. And then finally G is the third base, so with the third base, we look here, G, AUG, oh, it's methionine, the start codon. Excellent. All right, now that we know how to read this, let's look at some other examples. Let's say we have CCA. So we go C, C, A. Here we have a codon that codes for proline. Well, what if we have the codon CCU? They are different codons, but they code for the same amino acid. That's the idea of there being redundancy in the code. So you'll notice that there are quite a few examples of redundancy in the code, and what that does is help ensure that if there are mutations, let's say we've got CGU, oops, if there's a mutation and now it's CGC, well, despite the fact that there was a mutation, we still wind up with the same amino acid, and the functional protein is not in any way, shape, or form harmed. So we have this wiggle room by looking at the third letter. Notice how these are grouped by the third letter. So the first 2 bases of a codon will be the same, but you can have some wiggle room here in that third base. And again, what that does is help ensure there is some redundancy in the code so that if some mutations arise, it has minimal impact on the cell. The other idea is that there is no ambiguity. What is meant by that is simply that the codon CCA always codes for proline. It's not like CCA would code for serine or leucine. This particular codon, each particular codon only codes for one particular amino acid. All right, let's jump into section 17.2, looking at the actual process of transcription. Now we know that if you're going to use template DNA to make a replicated strand of DNA, a new strand of DNA, we use DNA polymerase. Well, it stands to reason that if we're going to use template DNA to make some RNA, we would use another enzyme called RNA polymerase. So RNA polymerase is simply the enzyme that pulls apart the strands of DNA and then synthesizes the new strand of messenger RNA. And again, the important rule to keep track of here is that U replaces T. Let's see. Now, transcription occurs in 3 stages. There's initiation, so we start to make the new chain. Elongation, so we make the actual transcript. And then termination, where we stop making the RNA molecule. So this is sort of a visual depiction from the book illustrating the events. So here's our double-stranded DNA, and here's the polymerase. So the polymerase attaches to the DNA, and it separates the strands. That's the initiation part here. So we attach the DNA, separate strands. Then they start to actually, RNA polymerase starts to actually build the mRNA, or the RNA transcript, or primary transcript. That is the elongation phase. Then RNA polymerase is going to hit a sequence on the DNA strand that causes the RNA to fall off, which we see here, and that refers to termination. So you start the process, you make the RNA molecule, and then we stop the process. This does a nice job of sort of illustrating how you're adding the nucleotides AUCG. Oh, one other thing, upstream and downstream. The production of the mRNA is directional. Obviously, we go from a 5' to a 3' end. The work that has already been done, or prior to the transcription, is referred to as upstream. The segments of DNA after this point of transcription is referred to as downstream. So when we talk about upstream, downstream, we're talking about before transcription, the point of transcription, or after the point of transcription. All right, so the parts involved in this process. Promoters, they encourage, right? A promoter will encourage or try to gain publicity for a boxing match. Well, promoters encourage RNA polymerase to attach to the DNA. So if we look at this original DNA strand here, notice how it's sort of green. That green area is the promoter. That is the area that encourages, if you want to think of it that way, encourages the messenger RNA, the RNA polymerase to attach to the DNA. The terminator, sounds intimidating, but the whole idea of the terminator is that it provides the signal, or the sequence of bases, that indicate to the messenger RNA to stop production of the RNA sequence. And transcription unit, all they're saying is it's the DNA that gets transcribed. So in the image here, they show the transcription unit. Again, that's only just the DNA that gets used to make the RNA transcript. All right, lots of fancy words, basic, simple ideas. Transcription factors. They help RNA polymerase attach to the DNA. If we look here, here are these transcription factors. They're just proteins. They are proteins that help RNA polymerase attach to the DNA. The way to think about this is like an adapter for an iPod. If you have like a clock radio and there are different adapters that can be used to attach different types of iPods or MP3 players, that's what these transcriptions serve as. They simply act as adapters that allow RNA polymerase to attach to the DNA. Now, when these transcription factors and these proteins combine with the RNA, it makes what is referred to as the transcription initiation complex. Ooh, big fancy term. All it's saying is that it's the proteins and enzyme that are used to begin transcription. So again, big fancy term, just saying it's the adapter for the RNA, and then the RNA itself, the enzyme itself, attaches to the promoter region. Okay. Last thing to mention here, the Tata box. Now, this is just a sequence of DNA, obviously T's and A's, that are recognized by these transcription factors. So this is at the beginning of every section of DNA that's going to be transcribed. You have this particular sequence. And again, the transcription factors attached there, act as the adapter, and then the RNA polymerase, the enzyme, attaches to create this transcription complex, and boom, you're off and running. All right, let's pause there.